In Chapter 6, we are going to be looking at different types of cell division. And also in this chapter, cell division is related to cancer, so I will also talk about how we can look at when cell division goes wrong and how that will form tumors. In your book, they talk about a story looking at chemicals from everyday products and what kind of health risks they pose for cells in relation to these different types of cell division. Cells will divide for two reasons. One reason is to reproduce an organism. So some types of cell division are used for asexual reproduction, and we'll see a type of cell division used for sexual reproduction. In addition to reproducing, cells will also go through division to grow and to repair cells. And this happens in multicellular organisms like humans, for example. One type of cell division that we're not going to spend a lot of time on is called binary fission. This is an example of asexual reproduction. This is a type of reproduction that we find in prokaryotic organisms, which are the bacteria and the archaea. We will also find this type of asexual reproduction in some single-celled protists that have eukaryotic cells. So how this works is we start off with a parent cell. The first thing that occurs is in that parent cell, it needs to replicate its DNA. So this is step one. That DNA will be replicated. It will also be segregated or separated apart. So we started off with one DNA chromosome, and now after DNA replication, the cell has two identical DNA chromosomes. Once that DNA is replicated, the cell will divide in half. So step number 2 is cell division. So what happens is the cell will create a cell membrane and a cell wall in between our two sets of DNA. Those two cells will then physically separate from each other, and we end up with two daughter cells that are genetically identical to each other. So this binary fission, we start with one cell, we end up with two identical cells. In eukaryotic organisms, cell division is a little more complex. So that binary fission occurred mostly in prokaryotic organisms that lack a nucleus and organelles, whereas eukaryotic organisms, they have a lot of organelles in them, they have a nucleus, and they have a lot more DNA. Many prokaryotic cells, like bacteria, have one circular chromosome, whereas eukaryotic cells, for example, in humans, we have 46 chromosomes. So you can see there's a lot more DNA that needs to be replicated, that needs to be split into two cells or into more cells. So eukaryotic cells, they go through a life cycle called the cell cycle, and this is a sequence of events that makes up the life of a typical eukaryotic cell. There are two main stages in the cell cycle of eukaryotic cells. The first stage is interphase. This is where the cell will spend approximately 90% of its life, and it's also when the cell just does what it normally does. So if a cell is a nerve cell, it will behave like a nerve cell and send electrical impulses. If the cell is a muscle cell, it will help with movement. During interphase, the cell will normally do its function. It can also get ready to go through the different types of cell division. So this interphase, because it makes up 90% of the cell's life, it's further divided into three parts. The first phase or first part is the G1 phase, and that stands for the gap 1 or growth 1. This is the longest phase, and this is where the cell will decide if it wants to divide or not. If it decides to go through division, it will get ready to replicate all of its DNA. If a cell decides not to go through cell division, the cell can enter into a side cycle referred to as the G0 cycle or G0 phase. This is an arrested phase or a non-replicating phase. So many of our adult cells will enter the G0 phase and they will not replicate. So again, this is where a cell will go if it decides not to go through cell division. It'll go off to this little side cycle. So some cells will spend their time in the G0 phase, and they can always enter back into the G1 phase and decide to go through division at a later time. So this G1 phase, if the cell does decide to go through cell division, then the cell will get ready for DNA replication. Once the cell is ready to replicate all of its DNA, it will enter into the next phase called the S phase, and this stands for synthesis. Specifically, this is where the cell is going to synthesize or replicate its DNA. So by the time we get to the end of the S phase, the cell will have twice as much DNA as in the normal cell that was in the G1 phase or in the G0 phase. Once all that DNA is replicated, the cell will enter into the G2 phase, which stands for gap 2 or growth 2, and this is where the cell will get ready to go through cell division. So again, this is all part of interphase, so the cell getting ready to go through cellular division. In that S phase, when the DNA is replicated, all of the chromosomes are copied and they're condensed down to prepare for cell division. So when a chromosome is replicated, it will look like a little X over here. And each of those arms on this X is called a sister chromatid. And these two sister chromatids are held together in a centromere. So during G1 and G0, our chromosomes look like a single strand right here. During that S phase, we end up with two identical strands that are attached to each other, and this is still referred to as a chromosome because those two sister chromatids are attached. Then they will condense down and we can see them a little bit more clearly. We have two sides, two sister chromatids attached together. Those sister chromatids are identical to each other. Once the cell goes through interphase and it has decided to go through cell division, it replicates all of its DNA, it makes sure that there's enough nutrients available, the cell will enter into the next stage, which is cell division. This is the last stage in a cell's life cycle, and this is when that DNA that was duplicated during interphase, specifically the S phase, this DNA is divided into daughter cells. And the first type of cell division we're going to look at, it's going to be mitosis, so that one cell is going to divide into two cells. So there are two types of cell division that can happen during this cell division part of the cell cycle. One is called mitosis. So this is a mitotic division that consists of mitosis, which is followed by cytokinesis. This type of cell division is used for asexual reproduction, and it is also used for growth and repair. In multicellular organisms. And mitosis is going to produce two identical daughter cells after we are done with this division. The second type of cell division is called meiosis. So this is a meiotic division that consists of two parts. There's meiosis 1 and meiosis 2, which are in turn followed by cytokinesis 1 and cytokinesis 2. This is a type of sexual cell division, so it is very specialized and only happens in certain organs in sexually reproducing species. And this type of cell division is going to produce the sperm and egg cells, which are the gametes. These gametes will only have half as much DNA as the mother cell. So from meiosis, this is going to produce 4 non-identical daughter cells. We're going to be looking at mitosis first. Mitosis consists of 4 main phases of the DNA process. So there's interphase, which we talked about. This is before the actual cell division event starts. That's where all of that DNA has been replicated. The chromosomes start to condense down. Those chromosomes will then enter into prophase. We'll have metaphase, anaphase, and telophase. Once you get to telophase, that'll be followed by cytokinesis, which is where the cell will physically separate to create two cells. So here's looking at mitosis. So we start off with interphase, highlighted in the blue. So again, this is where all of our DNA has been replicated. It starts to condense down. In addition, the nuclear envelope that surrounds all that DNA will start to break apart. Once that nuclear envelope breaks apart, the cell will enter into prophase. So prophase, we have 4 chromosomes in our cell here. So during prophase, those chromosomes continue to condense down, and we will have what's called a mitotic spindle set up. We'll end up with 2 poles. So in this case, we have one on the left, and we have another pole on the right. Those 2 poles send out spindle fibers that attach to the chromosomes. Those spindle fibers will pull back and forth, kind of like tug of war, until all the chromosomes are lined up in the middle of the cell. And this is when the cell is said to be in metaphase. After those chromosomes are lined up in the middle, the cell will do anaphase, where the 2 sister chromatids are separated from each other. So all of the arms on the left get pulled to the left. All of the arms that were on the right of the chromosome get pulled to the right. After those chromosomes get pulled to their respective poles, we have a nuclear envelope that will reform around the 4 chromosomes on the left, and then a second nuclear envelope will reform around the chromosomes on the right. This is telophase, when those nuclear envelopes are reforming. The cell will also go through cytokinesis, which is where those 2 cells will physically separate from each other. So this cell cycle that the cell will go through during its lifetime, there are certain checkpoints that the cell has to pass to continue on to the next step of the cycle. So these checkpoints are really important because they are going to check to make sure that the cell is good and that we can allow it to continue to go through cell division. So one checkpoint is called the G1 checkpoint, which happens during the G1 phase of interphase. So the cell will stop here, and the body will check if the cell is too small. It'll check to see if the nutrient supply is inadequate. It will check the DNA's damaged. If anything is wrong with the cell, it's going to stop the cell cycle and prevent it from going on. If the cell is big enough, if there's enough nutrients, if the DNA is fine, then the cell can pass the checkpoint and continue on. In this case, we'll go past checkpoint 1, go through DNA synthesis, that S phase, continue on until it gets to the G2 checkpoint. The G2 checkpoint, the cell will stop here if, again, the cell is too small, if the nutrient supply is inadequate, if the cell's DNA is too damaged, or if the chromosome duplication was incomplete. So this is another checkpoint to make sure that the cell is good, the DNA has integrity, everything was replicated properly. So this is really important because these checkpoints will stop cells from going through division if there's something wrong with them. If these checkpoints are not in place, or if something happens and the cell gets past the checkpoint and it's not supposed to, it could lead to uncontrolled cell division. It could form a tumor. So a tumor is when we have a cell mass that's created by rapid runaway cell division that has not been stopped by these checkpoints. And tumors, you can have 2 different types of tumors. So a tumor cell can be benign, and this is where the cell remains confined in one site. A lot of times that tumor cells will be in a capsule, and we can surgically remove those tumors. So this is not necessarily a good thing, but a benign tumor is a much better diagnosis compared to if you were told a tumor is malignant. So malignant, this is a very bad term in relation to tumors and cancer. So tumor cells are malignant when the doctor notices that the cell has become anchorage independent. So usually cells are firmly anchored in one place, and they will stop dividing if they become detached from their surroundings. But tumor cells, if they become detached from their surroundings, they will still continue to divide uncontrollably. So this is a bad thing. It signals that those cells, there's something wrong with them. They're passing those checkpoints. Their DNA could be damaged. It could be causing harm to that individual. In addition to looking at that anchorage independent characteristic, they also look to see if the cells have broken away and invaded other tissues. And this is called metastasis. If you have known anybody that has had tumors or cancer, metastasis is pretty bad. It's kind of a detrimental diagnosis. So this means that tumors have been able to spread from one organ to another, or it's spreading through the body. These cells will also promote angiogenesis. They will form their new blood vessels so that they're able to get more energy and more nutrients from the body. So these tumor cells really want to divide very quickly. They want to take over the body. They want to cause a lot of damage. So these malignant tumors, these are ones that we have to more aggressively treat. They could use surgery on some of the tumors, but a lot of times when it starts to metastasize or move, they will go into chemotherapy or they'll start to use other forms of treatment. So this is really interesting. So this is when good cells go bad. So it's really important that we understand mitosis and how it happens. And understanding mitosis and how it happens is really key to understanding how we can better treat cancer. So right now, once individuals get cancer, we try to make it go away. But if we really understand mitosis, and there's still a lot we don't understand about mitosis and DNA damage and those checkpoints, if we understand that, we will actually be better able to stop cancer from forming in the first place. So it's really interesting or important to know what happens when cells do what they're supposed to do, and then also what happens when cells go bad, or what happens when they go out of control. So that was looking at mitosis. So again, that's a form of asexual reproduction. It's also used for growth and repair. Our other type of cell division is meiosis or meiosis. So this is a type of sexual cell division where the gametes, the egg and the sperm cells are created. So meiosis or meiosis, depending on how you want to pronounce it, is a type of cell division used for sexual reproduction to produce gametes. These gametes are daughter cells that have half as much DNA as their original parent cell. So this is really important to understanding our life cycle, or how we can pass on genetic information from parent to offspring, and looking at that sexual reproduction. So these gametes that have half the number of chromosomes are called haploid cells. So these again are the sperm and egg cells. If we compare that to the majority of our cells, which are called somatic cells, most of our cells, like skin cells, muscle cells, nerve cells, they are somatic cells. They have the normal number of chromosomes, so they're referred to as diploid cells. And all of these diploid cells that we have are genetically identical to one another. So they have the same DNA as their parent cells. So these somatic cells are produced through that mitosis that we just looked at, not through the cell division that we're going to look at now, which is meiosis. So a couple of new terms here that we're going to need in order to understand meiosis. So we have diploid cells, which have all the chromosomes, and then we're going to have haploid cells, which have half the number of chromosomes. So these gametes, the egg and the sperm cells that are produced, they are used in sexual reproduction. So these egg and sperm cells can go through fertilization to produce a zygote, which will grow into the next generation, the offspring. So just to point out here, we're going to look at humans. So in normal humans, our somatic cells have 46 chromosomes. So the biological mother has 46 chromosomes, the biological father has 46 chromosomes. We have specialized organs, so females have ovaries, and males have the testes that undergo meiosis to produce the gametes. These gametes that are produced through meiosis end up being haploid, which means they have half the chromosome number. So the egg cells produced by the biological mom have 23 chromosomes. The sperm cells produced by the biological father have 23 chromosomes. The egg and the sperm cell will go through fertilization. We'll add 23 plus 23. We get back up to 46 in our zygote. So that fertilization process, our two haploid gametes that fused together are going to create a cell called a zygote that we're back up to our original 46 chromosomes. That zygote, that one cell that is formed from fertilization, it'll go through mitosis, so it'll go through growth to produce a new organism. So zygote, this is a single diploid cell. It contains all the DNA, so it has all 46 chromosomes. And it's going to undergo mitosis to form a new individual. So that zygote will divide and divide and divide to create a fetus. We're going to look at meiosis 1 first. So one thing to remember is that our DNA has already been replicated before the cell division begins, and this happens during that S phase of interphase. So these chromosomes that have been replicated, they contain the two sister chromatids, so they have a left arm and a right arm. These chromosomes are also found in homologous pairs. So I've mentioned that we have 46 total chromosomes. Those 46 chromosomes can be lined up in homologous pairs. So you can also say that we have 23 pairs of chromosomes. So these homologous pairs, this is important because this is what we're going to pull apart in meiosis 1. So in meiosis 1, we know that the cell already has twice as much DNA available because it was duplicated in interphase. This division is going to separate these homologous chromosome pairs from each other. So homologous chromosomes, here we have a pair. So what's going to happen is this pair of chromosomes are going to be pulled apart. So this chromosome on the left will be put in one cell. The chromosome on the right is going to be pulled to the other side and put into a second cell. In addition, cytoplasm is going to be separated to create two cells. So this meiosis 1 is referred to as a reduction event because our cells go from being diploid, or having 46 total or 23 pairs of chromosomes, to being haploid, or only having 23 total chromosomes. And that's because those homologous pairs have been separated from each other. So if we look at this example here, looking at meiosis 1, which again is this reduction division, in this cell we have 4 chromosomes, starting on the left. We have 2 large chromosomes and 2 small chromosomes. Those 2 large chromosomes are a homologous pair. The 2 small chromosomes are a homologous pair. So in this cell, we can say we have 4 total chromosomes, or 2 pairs. Those homologous pairs line up in the middle of the cell during metaphase 1. So we have our 2 large chromosomes at the top, the 2 small chromosomes at the bottom of the cell. The chromosomes on the left will get pulled to the left. The chromosomes on the right will get pulled to the right. So now we end up with 2 cells that each have 1 large chromosome and 1 small chromosome. So just to point out again, we started off with a cell that had 4 total chromosomes. Now we have 2 cells that each have 2 chromosomes. So remember, we're going from a cell that is diploid to cells that are haploid or have half the number of chromosomes. Those cells that we just created will then proceed through meiosis II. So in this case, DNA is not replicated again. We don't have another interphase that occurs. Those cells coming out of meiosis I will go directly into meiosis II. So the cells have half the chromosome number. They are haploid. And this division is going to separate our sister chromatids to create 4 daughter cells. So if you remember, on our chromosomes, they look like little X's. The left arm is a chromatid, and the right arm is another chromatid, and those are sister chromatids. So we're going to be pulling those 2 chromatids apart. The cells are still haploid because those sister chromatids have been separated. So we're going to have cells that have 2 chromosomes in them. So here we can see meiosis II. So we have our cells that are haploid. They have 1 big chromosome and 1 little chromosome. Those chromosomes line up in the middle. The sister chromatids get pulled apart from each other. We end up with 4 daughter cells. These 4 daughter cells are not genetically identical to each other. So this is going to be really important. And we can see that here. So 2 of our cells have a big red chromosome and a small purple. Our other cells have a big purple and a small red. So you have different combinations of chromosomes that occur in these gametes. So this genetic diversity that I just pointed out, this is really important because it will help with sexual reproduction. So the whole purpose of reproducing sexually is to increase genetic diversity, increase your chances of having very genetically diverse offspring. So there's a couple of things that contribute to this genetic diversity. One is that half of an organism's DNA comes from different parents. So in you, half of your DNA came from your biological mom, the other half came from your biological dad. So this makes you very genetically unique. You are not your mom, you're not your biological dad, you are your own person, you're a combination of both. Meiosis and fertilization can increase this genetic variation. So one of them is that fertilization is very random unless you go into an in vitro fertilization clinic. Natural conception, a random sperm cell will fertilize the available egg. So fertilization is very random. In addition, the process of meiosis can increase genetic diversity, and we'll look at 2 ways that genetic diversity can be increased. Another big thing that I pointed out is that meiosis, because it cuts the chromosome number in half, our chromosome number can remain constant from one generation to the next. So remember that both parents have 46 chromosomes. During meiosis, that chromosome number is cut in half. In those gametes. During fertilization, we add those chromosomes back together. We get back up to 46. Right there. So this is also really important to half that chromosome number during meiosis. Otherwise, with each generation, our chromosome number would double, which pretty much no species could survive that. Just due to the size of the cells, we can only fit so much DNA in the cells. So we have to half it first and then get back up to that original number. So back to this genetic diversity. Genetic diversity is really important. So there's a couple of ways that meiosis will increase genetic diversity. One of them is called crossing over. So this is when there is a physical exchange of chromosome segments between non-sister chromatids in your homologous chromosome pair. So this occurs during meiosis 1. So we will take a look at this. If we have a cell up here, it has a homologous pair of chromosomes, so they're the same size, same shape, they carry similar genes. What happens during prophase 1 is that these homologous chromosomes, they get so close to each other that parts of them will physically cross over with each other and a physical exchange will occur. So if you look very closely, this pink reddish chromosome on the left now has a little purple part to it, and the purple chromosome now has a little pink part to it. So when those sister chromatids are pulled apart, we can get a lot of diversity. So we can have a cell that has one pink chromosome, a cell that has a mostly pink with a little bit of purple, one cell that has mostly purple with a little bit of pink, and a fourth cell that has an all purple chromosome. So here we can see there's a lot of diversity that can occur. So crossing over can happen more than just once, and it can also happen on all of those homologous pairs. So remember, we have 23 homologous pairs of chromosomes. So crossing over is going to create new combinations of DNA, so that physical exchange. In addition, these chromosomes are going to be randomly distributed into the daughter cells. This is called independent assortment. So the random distribution of those homologous chromosomes into daughter cells, this also occurs during meiosis 1. So I'll show you what that looks like. So here we have this independent assortment. So we have two different ways that a cell could go through meiosis. We have option 1 over here, where similar colored chromosomes get pulled apart together. So our two pink chromosomes get pulled into the left. So we end up with gametes that have pink chromosomes. And the purple ones get pulled to the right, so the gametes have purple. Another option that could occur is over here on option B. So here our homologous chromosomes are set up differently, and our big pink chromosome gets put into a cell with the little purple, which will create different combinations. The big purple chromosome gets put into a cell with the little pink chromosome, so we get even more combinations. So you can see all these different possible gametes at the very end. You can have a lot of different combinations. And these chromosomes, they really are lined up randomly. So similar chromosomes are not going to be put into a cell together. It's really, really random. So this, again, increases genetic variation, which will increase genetic variation of the offspring. So we looked at our two different types of cell division. We looked at mitosis. We also looked at meiosis. So a good thing to kind of do is to work through those two types of cell division independently to understand them, and then kind of go back and compare them to each other. So they look very similar, but there are some major differences between mitosis and meiosis.